you very much. What I'd like to talk about in the last few 10 minutes or so is CRISPR. So you also study CRISPR, and I saw recently that you used CRISPR on a chip to do like an SMP check to find whether somebody had a disease or not. Could you talk a little bit about how that works and why that's so special?
So first of all, I want to say that it is work of really incredibly talented young woman scientist, Professor Kiana Aran, who did quick and successful postdoctoral work in the Conboy laboratory. And so she learned about CRISPR. It was like a big deal about CRISPR back then with Jennifer Doudna and CRISPR discovery. And she had this great idea to use CRISPR for diagnostic. And so she came to my office and we started discussing what would be the best way to adapt CRISPR-Cas9 for diagnostic of disease. And so it was really her idea that kind of just helped with collaboratively from my perspective and my knowledge. And so then she started her laboratory at her graduate institute, and we continued to collaborate. We are each other's favorite collaborator, in fact. And that approach to identify single nucleotide polymorphism or point mutations were particularly challenging with CRISPR, because you are looking at big chunks of genome, but you want to distinguish difference in one single base pair. So basically looking at a lot of text and trying to find one typo in one letter. And so that requires...
So it's not just the needle in the haystack, it's like the needle in Central Europe.
Exactly, something like that. But it is very, very important. And so we through continuous discussions, how to do it, how to make this approach work, we finally made it work. But it was really Kiana's laboratory, her group and her company efforts that made the device and basically made it from idea to actually something that is physically, you can physically touch it and it works. And the key point of the discovery is that now we can, without gene amplification or PCR, bulky instruments, very quickly identify mutations in cells, but also what is called genetic drift. So when we grow old, there are certain changes in our genome which accumulate. So you talked about burden of senescent cells, but there's also burden of DNA damage and mutations. And it would be incredibly important to be able to profile for these mutations from, for example, peripheral blood samples and link them to disease diagnostic and prognosis and treatments. Can we monitor aging very quickly through CRISPR single nucleotide polymorphisms? Can we apply this device to basically screen from peripheral blood samples propensity to develop certain diseases?
So at the moment, it looks for a particular disease, is my understanding. So, but you would hope to create a more generalized system that would look for wider set of diseases or a wider set of changes.
Yeah, so actually for the moment,
look for, right?
Yes, so it does look for particular disease. You can adapt it to any disease that you want. For example, in our paper, they looked at sickle cell anemia as the main concept, and then reviewers asked, can you look at something else? And we had samples to look at the ALS patients, Lou Gehrig disease. So the same system was successfully applied to that. But in order to do it, yes, it is a candidate approach. So you have to postulate which genes are likely to be mutated if you are looking at the aging or profiling, or which genes which mutated will have higher propensity for cancer development. But those are already known, right? This is the whole field of bioinformatics, and every single gene has been annotated and identified. So it is not really a scientific problem, it's just kind of the number problem. And then additional way to use this is to screen without using cells or animals for the best CRISPR machinery. There are many people who want to use CRISPR for gene editing, but they don't know what particular enzyme to use and what particular guide RNA to use. So Kiana's system allows to screen all of that in vitro in a very quick way. So that just empowers the field of CRISPR diagnostic and CRISPR editing.
Okay, so speaking of CRISPR editing, so it was in 2017, but you were co-author on a paper about CRISPR Gold. I just wondered, because that seemed so cool, being able to do gene editing without using a virus to put the gene in. Has there been any progress on that? Because like I say, it seems really cool.
Mike
You need to talk to Conboy.
Yeah. So Mike, in fact, made this term CRISPR Gold. He is excellent. Mike Conboy, he is excellent in giving terms to scientific phenomena like the best. So CRISPR Gold, right? Because it's gold nanoparticle, but it's the best. It's gold is what we associate with high quality, right? So CRISPR Gold. So that was our attempt to do something which is called homologous DNA repair, which we would need to have to actually correct mutations, not to cause mutations, but to correct them, the healthy gene sequence. And that work now shifted to the former students who was supervised by Irina and Michael laboratory, and we co-supervised him for that project for a couple of years, Conboy Lee. So he started company GeneEdit, and you need to talk to him. He will tell you all of the next steps and the progress in this direction.
We don't know.
Okay.
No, I know a little bit, but but not as
Okay. Yeah. No,
it's not a secret, by the way. They have good website there.
Okay. I will look him up. Yes.
Yeah.
But anyway, because it does seem like really interesting to be able to inject something.
He was on Forbes. He's a young person, very talented. So I think it is
30 under 30 or whatever it was, or some other thing.
30. No, he's not under 30, but maybe like something under 40. Or maybe he's under 30. I don't know. But you can find him. His name is Conboy Lee. It's actually spelled Conboy, Conboy Lee, and he is CEO of a company GeneEdit.
Okay, excellent. I will do that. Okay, so thank you so much for both of your time today and for
You're welcome,
your explanations.
Awesome. Thank you so much for your interest.
Okay, thank you so much.
I hope that you found the video informative. Please do hit the thumbs up button, subscribe to our channel, and hit the bell button for any new video release notifications. Thank you so much for your kind support. I wish you all well and will speak to you again soon.